Welcome to Pharmaceutical Technologies Podcast, Biopharma Frontiers, navigating innovation and regulation in cell and gene therapy. This podcast is brought to you by Solvias. Solvias provides CMC analytics to advance your path to commercialization. To find out more, please go to solvias.com. And now, here's your host for this podcast, the Senior Editor, Special Projects for Pharmaceutical Technology, Megan Manzano.
Hello, everyone. This is Megan Manzano, Senior Editor of Special Projects for Pharmaceutical Technology, and I'm here with Daniel Galbraith, Chief Scientific Officer of Solvias. How are you today, Daniel?
I'm well, thank you.
Thank you so much for joining me today. So today we'll be talking about advancements in the evolving world of cell and gene therapy analysis. So to get us started, Daniel, could you give us a little bit of background about Solvias and your role as Chief Scientific Officer?
Sure, thanks for the question. So Solvias has been in business for around 25 years. We're actually celebrating this milestone in September. We're established as a centre of excellence for analytical chemistry all those years ago, and we continue to be a preeminent CRO for understanding the chemistry of new drug entities. But over the years, we've increased in the diversity of the types of drugs, modalities that we test, and also expanded the technologies we use. Fast forwarding a little bit today, we offer a complete testing analytical package for small molecule and biologic drugs. And we've been really excited in the expansion that we've seen in our offering for the cell and gene therapy drug developers. Part of the expansion of Solvias has been acquisitions of some new services, and we have a team delivering expertise in cell-based potency assays and microbiology in France. And more recently, we have a team in Utrecht in the Netherlands who provides some of the best next generation sequence technology available on the market. And hot off the press was our announcement very recently of our new set in North Carolina in RTP, where we're currently building a 50,000 square foot cell and gene therapy centre of excellence, which offers dedicated services to the market in North America for our clients. Now, you asked about my role as a CSO. I feel I democratize science. We have a tremendous resource in our team of experts who provide technical and regulatory advice to drug developers at all stages of drug development and at many different modalities. So my role is to make sure the best of our science is in front of our customers and to make sure that the challenges that the drug developers face don't cause any delays to these drugs, really. I also have a role in understanding what are the new ideas and technology we need to support our customers and bring the voice of our customer back to our internal teams and to help with their challenges. It's a tremendously exciting time in drug development, and I really do enjoy this role.
Thank you so much, Daniel, for that. And that leads us very well into my next question for you, which is, with the work you have done for several cell and gene therapy companies, what are some of your key learnings from these experiences?
Thanks. The way we partner with our cell and gene therapy clients is really important to our success, as well as ensuring that their supply chain is effective. And testing cell and gene therapy drugs presents some new challenges as we move through the drug development timeline. Some of the testing requirements for CGT drugs are not very complicated. We still measure things like pH or subvisible particles, and these tests can be thought of as being relatively simple to do. Some tests, such as sterility, again, are thought of as being quite simple to do. But what the difficulty is with these tests is that the traditional methods take a long time. So, for example, sterility, as I mentioned, takes 14 days in culture to determine the absence of bacteria in a sample. Now, that becomes a problem when a lot of our drugs that we test in the cell and gene therapy space have a very short shelf life. And our challenge has been to find alternative methods to establish the safety of these drugs. We need to show regulatory acceptance of new technologies, validate that there's no loss of sensitivity, specificity, and robustness. And some of these can take quite a long time for us to actually establish for our customers. So we've got a plan in place to actually make sure we've already taken account of these. Some of the testing that we perform for our cell and gene therapy customers by their nature, are a little bit more challenging. The best example of these are the potency assays. Each cell and gene therapy drug developer is unique, and we need to be able to support each project with specific assays that are developed, qualified, and validated to meet regulatory standards. And as the number of these drugs grow, so the burden of this testing rests on our shoulders. Our new facility in RTP will help resolve the capacity issues for the next few years, but we need to keep building and keep growing our capacity. When we start working with a new project, we also usually start off with a very small number of samples to be tested. Early stage clinical trials tend to be small. Each of the sample is tested for a number of different tests. And as the clinical trials move on, we have to expand the number of samples that we test. And obviously, we need to make sure we have the capacity to do that. But it's only really when it comes to moving into the commercial stages of a drug where we actually move from maybe tens or hundreds of samples to maybe tens of thousands of samples. And then it becomes critically important that we have very in-depth conversations with our customers to make sure that we have the right capacity, the right supply chain, and the right timelines to make sure that when they're in the commercial stages, they're ready to go and able to treat as many patients as they possibly can.
The next question I have for you is, could you tell us a bit more about your partnership with Vertex Pharmaceuticals?
So we were tremendously grateful for the team at Vertex to allow us to publish this press release describing the relationship we have. And we're very excited at being just a small part of the supply chain they have of bringing this new drug to market. We've been working with the team at Vertex for many years, developing analytical testing to release their drug and treat sickle cell and beta thalassemia. Now, our planning with the senior team at Vertex established a dedicated space in our labs in France and in Switzerland to ensure we have capacity both in terms of the manpower and the laboratory space. We've seen the first of these patients treated with this drug after approval, and we're actually on a very good trajectory to support them through the commercialization and the increase in number of samples. And it's quite exciting to be on that path with them.
It's well known that cell and gene therapy is a dynamic field with changing regulatory requirements. How do you see the regulatory landscape for these therapeutics evolve?
Short term, there's probably two pieces of regulatory guidance that have changed or are about to change that are going to really impact us. The ICH Q5A guideline was first put in place in the mid-1990s, and it was revised towards the end of 2023. The update has widened the guidance in scope, and it now includes cell and gene therapy drugs. And it's brought new technologies into the fold as being acceptable for testing these packages. And this is critically important for cell and gene therapy manufacturers. For me, the revision of the guidance was long overdue. However, it's great for it to be in place now. And as I mentioned, it's bringing these new technologies in. The importance being that the new technologies can very much speed up the length of time it takes to actually do the testing. And that is critical for, as I mentioned earlier, some cell and gene therapy products as they have such a short shelf life. We're now in a process of the guidance is in place, and we're now converting our customers to these newer methods. Everyone I've spoken to is very excited that this is in place, really so that they can release their products much safer and much faster. One important thing to sort of include in this is that they have the new guidelines has made it possible that we no longer need to use animal testing in the biosafety aspects of these drugs, which is something I'm very pleased about, that we've been able to move that forward. So that's now in place, and as I mentioned, we're moving forward with that. The next guideline that is potentially going to sort of change our industry is the draft guideline that was issued by the FDA. This was towards, again, the end of last year, still in draft. And that was on the potency assurance for cell and gene therapy products. This revises a 2011 guidance. Now, It's a very long document, but it really is worth the effort of everyone to read it if you are involved in releasing cell and gene therapy products. The guidance does propose significant changes to the premise of what we measure and how we measure the potency of these drugs, which is really very important. A few topics that are well thought through, in my opinion, are some of the issues of compliance, testing raw materials, critical quality attributes, and critical process parameters. And they also include some in-process controls are all highlighted and made us think really how we approach these assays. There's requirements for at least one bioassay, which wasn't really clear in the previous guidance, and that bioassay should focus on the mechanistic aspects of the drug. And of course, it suggests, again, we should avoid animal testing. One thing the guidance does help with additionally is how we should speak to the agency, the FDA in this case, on these issues, which I think is really useful. This hasn't really been very clear in the past. So those are the two guidances that are, regulatory guidances that are really going to be critically important that people understand and take action on in the very near future.
Diving into change a little more, how do you see the future of the cell and gene therapy market space evolving?
Yes, with 3 molecules already approved this year and a further 4 likely to be approved by the FDA in 2024, I think this is going to be a bumper year for this modality. Cell and gene therapy treatments still focus for the main on incurable genetic disease. Many of these conditions really have a very small patient population, although sickle cell, with 100,000 people affected in the USA alone, shouldn't really be thought of as a small patient population. However, short term, I think the future will expand the number of these genetic diseases that will be treated with cell and gene therapy products. And I can predict there will be much more investment in this space. In a medium term, we definitely will start looking at gene therapy as a solution for treating other conditions aside from the single genetic conditions. And I think we'll see cell and gene therapy augmenting other therapeutic treatments in an effort to sort of cure disease. Long term, I think the majority of disease will be treated with some form of cell therapy and/or gene therapy at its core. Long term, the administration of pharmaceuticals will probably become a thing of the past, and we'll be looking really to manipulation of our bodies really to treat disease rather than looking for external pharmaceuticals. But that's a very long way off, and it's only my speculation on the future.
Great, thank you. So currently, the cost of cell and gene therapies are extremely high. Why is this the case, and how do you see this being managed in the future?
Yes, you make a very good point. Cell and gene therapy treatments are sometimes hundreds of thousands of dollars per patient in treatment, and it's clearly outside the ability of many healthcare agencies to afford. Most of these treatments are created and customized for each patient. Usually we use the patient's own cells. These are manipulated, cultured, and then given back to the patient. So these individual treatments such as these are expensive. Very little can be done for the product to reduce costs, due really just to the nature of that individual manufacture of the product. When we start to move away from patients' own cells, and we can actually use more generic treatments, allogeneic treatments as they're called, then we could be in a place where costs can be reduced. However, there really is a number of hurdles that we need to get through before we can make this the mainstay. Despite this, many drug developers are out there and they're progressing with these types of drugs. We're probably looking at a few years, maybe 4 or 5 years before we see these actually being as successful as the current crop of cell and gene therapy drugs. Now, alongside manufacturing costs, the costs of the QC testing that Solvias is involved in also is a consideration. As I mentioned, we have to develop unique tests for the products at present. Moving forward with the generic nature of these drugs, obviously we can reduce that cost. But we also have a lot of operational excellence strategies that we have in place to bring down the QC release costs of cell and gene therapy. And we're hoping to bring these much closer to what we see for traditional biologic drugs. However, to do this, we can't do it on our own. We need a great working relationship with the drug developers, but also the regulatory agencies play a crucial role in this as well.
Fantastic. Thank you for that. My other question for you here is, in your role as CSO, are there any particular exciting technologies that you see emerging?
Yeah, it's an interesting question, and you know, there's always new technologies and that are coming into place. One I'm going to pick out is really the technologies that we're seeing where we have the ability to understand what is happening in a single cell. So there are some technologies now where we have the potential to understand, you know, what of the proteome is changing within a cell. And I think when we start to understand this fundamental basis of what's going on in a cell, we really will enable us to understand and comprehend the biology of disease in the future. Just to use an example, cancer, everybody knows what cancer is, but essentially a cancer cells, they're mutating, they're changing all the time, and you have many different lineages that are unstable. Some are changing faster than others. If we have an ability to look at these different lineages, understand what's going on with them at the cellular level, we can then target how we treat that condition and you know, have a more holistic approach to that treatment if we really do understand what's happening with them all. With these newer technologies, if we can achieve that, I think that will bring a fundamental change to the way that we look at disease and the way that we treat disease.
Absolutely. All right. Well, I think you've given us such a wonderful amount of information today, Daniel. So thank you so much for joining us today.
No problem. Thank you for your questions.
This has been Megan Manzano, Senior Editor of Special Projects for Pharmaceutical Technology. Thank you, everyone, for listening.
You've been listening to Pharmaceutical Technologies Podcast, Biopharma Frontiers, Navigating Innovation and Regulation in Cell and Gene Therapy. This podcast is brought to you by Solvias. Solvias provides CMC analytics to advance your path to commercialization. To find out more, please go to solvias.com.